Hi, and good day to you. Welcome to the Cerebellar Seminar Series. Today, as our guest, we have Céline Auger, who received her PhD at the Max Planck Institute for Biophysical Chemistry at Göttingen, under the mentorship of Alain Marty. She studied inhibitory synaptic transmission between inner neurons of the molecular layer of the cerebellum. She was subsequently a Human Frontier Science Program postdoctoral fellow at the University College London in the lab of David Atwell, studying the recapture of synaptic glutamate by transporters at the climbing fiber synapse. Then she was a research scientist at the Brain Neurophysiology Lab at University of Paris, Descartes, working with David Ogden, and now is a co-team leader of the Cerebellar Neurophysiology Lab at University of Paris in France. And recently they discovered a lovely description of the granule cell inputs to the Purkinje cells, particularly differentiating between their inputs to the Purkinje cells before it becomes parallel fibers. And I'm happy that to have her explain that work and teach us about it today. Thank you for being with us, Céline.
Hello, everybody, and thank you, Reza, for the kind introduction and the invitation to present this work. Today, I will talk about a new form of associative plasticity that we characterize as the granule cell inputs to Purkinje cells. Oh no, this is not working. Here we go. So the Purkinje cell has a huge dendritic tree that's organized in the sagittal plane, so that's perpendicular to the parallel fibers, and it's a major site of integration of information in the cerebellum. And those huge dendrites, you can see, are covered in a very high number of spines, where parallel fibers and the granule cells form about 175,000 synapses on a single Purkinje cell. So those spines are equipped with ionotropic glutamate receptors of the AMPA type, and metabotropic glutamate receptors type 1, but no NMDA receptors. And they are a well-known site of synaptic plasticity, both LTD and LTP. By comparison, the climbing fiber input makes about 2,000 synapses on a Purkinje cell. So of this huge number of granule cell synapses, it turns out that a major proportion of those inputs are silent. In fact, there's no detectable activation of AMPA receptors or activation of synaptic currents. And this was indicated by work in vivo by Jontel and Eckroth. And this was shown in slices by paired recordings between granule cells and Purkinje cells by Aesop and Barber. And you can see that some of those connections show strictly no electrophysiological response, while others generate sizable responses. And 85% of the connections are thought to be silent. I should have activated my cursor, but no, never mind.
No.
Can you see my cursor? Because I should have...
Yes, we can see it.
Okay. So most of those are silent. Obviously, some of those granule cell synapses are not silent, and we showed that they can actually come in two versions, which are associated with different receptor signature. So for comparable stimulation and train stimulation of the inputs, some of those responses show a large initial response, facilitation, but quickly saturate, while others show very small responses that facilitate throughout. So these are currents recorded in different cells, and many of these are actually hybrid responses of the two. But the same is true at the level of a single Purkinje cell. And if we scan the dendritic tree of the Purkinje cells, we observe the two types of responses. And analysis using pharmacological compound actually showed that those two components are actually have different sensitivity and are linked to different functional involvement of the top auxiliary protein. So those different responses, if we use a similar stimulation intensity, will give different synaptic current, and they will also generate a different output pattern in the Purkinje cell, with here in this case a delayed and long-lasting burst, and in other cases a very rapid and response in very short burst of action potential. So I just quickly want to come back on the anatomy of the granule cell axon. They're shown here, for example, the granule cells are here. And you can see that the axon first rises through the Purkinje cell layer and into the molecular layer. And before it bifurcates at right angle to form the parallel fiber. Both portions of the axon, both ascending axon and parallel fiber, form synapses. But the ascending axon can only make contact with a couple of local Purkinje cells, while the parallel fibers can make synapses with hundreds of Purkinje cells. So they actually potentially distribute a different information. The ascending axon distributes a very local signals from local granule cells, while the parallel fibers will distribute a distal signal, which I call contextual, because it's going to be a very varied signal. So we were interested in those ascending axon synapses, so we simply recorded from Purkinje cell and stimulate them below. And we noticed that they had a different decay kinetics, which were fast, no facilitating component, no mGluR current, and they were also recruiting more inhibition. And the other interesting point is that Sims and Hartel had shown that they would seem to be resistant to plasticity. So they could show no LTP with well-known protocol, and no LTD either with conjunctive stimulation of the climbing fiber. So what's the interest of having those different types of responses? They can obviously generate different output patterns for the Purkinje cell. They can also be associated with different plasticity rules. What do the different synapses encode? Well, really, we don't actually know. And I would like to bring them back into the context of the cerebellum to finish that little introduction. So what does the cerebellum do? We think the cerebellum is required to correct and adapt a motor program during perturbation. For that, it receives motor information, sensory information, and also reward. And we think that it makes predictions of the consequences of actions and uses that to correct the actions. How does it do that? Well, again, we don't really know, but I think we have a good hint when we look at work done on cerebral light structures in electric fish. So quickly, I'll go through that. So the electric fish use both active and passive electrolocation to locate prey and see the world around them. And to do that, they have cerebellar-like structures that analyze the motor command and the sensory feedback in order to subtract the activity generated by their own movements, their own activity. And in this way, isolate the external signals of interest. So at the synaptic level, how does it work? And here I've taken the example of the electrosensory lobe of a marmoret fish. And that's taken from a review by Nat Sauter. So here, both the motor command to the electric organ, but also to the swimming muscles of the fish, are fed by the granule cells to the apical dendrites and synapses of the principal cells by parallel fibers. So these are parallel fibers. The electrosensory inputs are actually fed directly to the basal dendrites of those principal cells. Now, both signals can by themselves generate the activation of these cells, but if they're presented repetitively together, the parallel fiber synapses are modified following a precise plasticity rule, and they're modified in order to generate a negative image of the electrosensory signals. So in this way, the fish is capable of subtracting the activity linked, it's capable of subtracting all the consequences of its own activity and isolate external inputs. So in the cerebellum, we have no direct sensory inputs and no basal dendrites in the Purkinje cells. But also the granule cells are organized a little bit differently now. They're not located in lateral nuclei, but they're now sitting below the Purkinje cell. And they make these synapses with these two portions of the axons, the ascending axon and the parallel fibers. So it's inspired by this work that we actually asked whether the stimulation of those two types of granule cell inputs to the Purkinje cell could lead to plasticity and could participate in the computation made by the Purkinje cells. So very few technical points. We use patch clamp recording to record the synaptic currents in the Purkinje cells of the rat cerebellum. So we work around 3 weeks of age. In these experiments, we preserved inhibition and we used low internal chloride to mimic really the physiological gradient. And I will come back later on to the why it was important for us to preserve inhibition. We then used 2 stimulating electrodes, extracellular stimulation electrodes. So if we're recording new imaging, we're recording from that Purkinje cell. One stimulation electrode is positioned right below the Purkinje cell, and it'll stimulate not the Purkinje cell, so this we can avoid, but it'll stimulate parallel fiber, it'll stimulate the granule cells that are sitting right below in the axis of the Purkinje cell and that are going to make ascending axon synapses. And we use a second stimulating electrode positioned in the molecular layer. And here we're going to stimulate at a distance. We stimulate a beam of parallel fiber synapses here in this case. So the data, this is what the data look like. We're recording from a Purkinje cell and stimulating the 2 inputs are color-coded. So in blue for the ascending axon and orange for the parallel fibers. And we record the synaptic currents, synaptic current amplitude. And we use paired pulse because this is going to give us some idea whether there are some changes happening presynaptically. So the continuous lines show the EPSC or the synaptic currents in general, in blue for the ascending axon, in orange the parallel fibers. We then switch the recordings to current clamp and stimulate both inputs simultaneously using trains of stimulation. And we use this stimulation protocol because it's been shown to induce plasticity at parallel fibers, both in vivo and in vitro. We then switch the recording back to voltage clamp, and we follow the amplitude of the synaptic currents for half an hour. to see how it evolves. And dotted lines show the currents after a half an hour recording. So you can see here in this example that the ascending axon synaptic currents has increased, while at the parallel fibers, it has decreased.
Céline, may I ask a question?
Sure.
It wasn't clear to me, how does one stimulate the ascending axon, which of course is going to produce also activity in the parallel fiber, no? And how does one dissociate like antidromic activity from the parallel fiber down to the ascending axon?
It is rather, I would say it's statistical. When we are stimulating right below the Purkinje cell, most of these granule cells are going to make ascending axons. And the slice is not very thick. It's actually 300 microns thick. So it can also, you know, actually the dendrites of the Purkinje cells are arranged in a plane, and the ascending axon is going to arise in the same plane. So if we take granule cells that are located nearby the Purkinje cell dendrites, we are going to stimulate a good proportion of granule cells that make ascending axons that will come in contact. It doesn't mean that we are stimulating 100% ascending axon. There will surely be some parallel fiber synapses, and I would say they are proximal parallel fiber synapses in that case. They are not formed by the end of the parallel fibers, but this is not 100%. I would say we are statistically is going to be enriched in ascending axon. Same is true for the parallel fibers. Obviously, some of those fibers we stimulate can bifurcate at the right place and make an ascending axon that synapses onto that Purkinje cell. But statistically, when you know the length of the parallel fibers, so on average over 4 millimeters in an adult, statistically the chance they bifurcate at the right place in the 50 micron thick slice that defines the dendrite of a Purkinje cell, it's very small. It's actually on the order of 1 or 2%. So
Thank
here,
you.
It's not pure, but it's statistically strongly enriched in parallel fiber. Last thing I wanted to mention. So I told you that we've preserved inhibition, and here excitation is inward, inhibition is going to be outward, and the currents you see are actually the sum of both EPSCs and IPSC. There's excitation and inhibition. So some of the currents here look like they decay very quickly. That's because there is inhibition coming right after. And here sometimes they have strange kinetics. That's all down to inhibition. However, we've shown that because there's an extra delay, synaptic delay for inhibition, we actually have time to measure the EPSCs properly. And we can come back on that if you want later. So that's what the average data look like. And here, so we've normalized the amplitude of the synaptic EPSC, so the excitation, and it's normalized to the last 5 minutes of control. And you can see that following the stimulation protocol here during this gap, the amplitude of the EPSC, the ascending axon EPSC in blue, for the first and the second response is increased, and it's still increased, shows a long-term potentiation at the end of the 30 minutes recording. The parallel fiber EPSC for the first and second response here in orange, on the other hand, shows a depression. This graph shows the ascending axon amplitude relative to the parallel fibers. And you can see that following induction, the induction protocol, the ascending axon doubles relative to the parallel fibers, and this is stable over time. Finally, we can see that the pad pulse ratio decreases, but this effect is transient and disappears by the end of the experiment. So this long-term potentiation mostly isn't linked to presynaptic changes. So we see that we have a mechanism of long-term potentiation of one of the inputs, the ascending axon inputs, while we have a depression of the second input. Now you can also notice on this graph that during the control period, the amplitude of the synaptic currents actually decreases. And in fact, we observe a rundown of the currents. So we did a series of experiments just to quantify this rundown of the synaptic currents, where we do exactly the same experiment, except we have no stimulation protocol. And you can see that in this case, the amplitude of the EPSCs is on the order of 80% at the end of the experiment. But that rundown is affecting both inputs in the same way. It does not change the relative amplitude of the two inputs, and that it doesn't affect the pad pulse ratio either. So rundown cannot be mistaken for a long-term potentiation, cannot be mistaken for an increase. And if anything, we are actually estimating this potentiation. However, that rundown makes life more difficult when we look at depression of the synaptic currents. And that's for that reason we speak about the potentiation of the ascending axon. We will not discuss further actually that depression of the parallel fiber EPSCs. So next question was to see whether plasticity was actually just specific of the ascending axon inputs, or whether it was also a form of associative plasticity. So that means whether it required the conjunctive stimulation of the parallel fibers. So we did a series of experiments where we stimulated ascending axon inputs only or parallel fibers only. When we stimulate the ascending axons only, you can see that we observe strictly no LTP and no depression apart from the rundown. When we stimulate the parallel fiber inputs, the same, obviously, I want to say there's no effect on the ascending axon and no particular effect on the parallel fiber either. So we have a form of plasticity that's specific of the ascending axon inputs, but it's also associative. It requires the stimulation of the parallel fiber inputs as well. We then looked at the effect of relative timing of the stimulation of the two inputs. And here first, we delayed the stimulation of the ascending axon by 150 milliseconds. And you can see that in this case, the response is very different. We completely lose the ascending axon LTP, which transforms into a deep depression of the EPSCs. A depression that's very similar to what we observe at parallel fibers. So there's no real change of relative amplitude of the inputs, although with time here the value increases because the currents are small, but they both getting even smaller, so it gets more noisy. When we now shift the stimulation of the ascending axon earlier, so that's before the parallel fiber, this time there's still some level of potentiation of the inputs, but nothing significant. Interestingly, in this case, the amplitude of the parallel fiber EPSCs is not depressed. So when stimulation of the two inputs is simultaneous, we have an LTP of the ascending axon. When ascending axon stimulation is delayed, we have an LTD. And when it comes early, there's not much effect on the ascending axon inputs. So plasticity is timing dependent, and this is kind of what we expect for a form of associative plasticity. We then wanted to compare this form of plasticity to what's been described at the parallel fibers, both LTP and LTD, and we wanted to compare molecular pathways that are involved. So LTP and LTD of the parallel fibers require a rise of the intracellular calcium concentration, with LTP triggered at lower concentration and LTD at higher concentration. And then there are two different pathways, molecular pathways that have been involved. One is activation of NMDA receptors, which triggers the production of NO, and glutamate release, presynaptic glutamate release has been shown to activate presynaptic NMDA receptors, which trigger calcium increase, NO synthase activation, and NO production. NO can then diffuse across membranes and activate guanylate cyclase postsynaptically. And this pathway, together with a rise of intracellular calcium, will trigger plasticity. The other pathway that's been described in LTD at parallel fiber is the activation of the mGluR1 receptor. And we know that mGluR1 is present on the postsynaptic spines. It can also be activated by glutamate release at the synapse. And it will converge onto an increase of the postsynaptic calcium concentration because it activates phospholipase C, but also a tryptide channel that's permeable to calcium. And it can also modulate voltage-gated calcium channels, which feed onto the calcium concentration. mGluR1 is also activates DAG lipase, which will trigger the release of endocannabinoids. So we tested the involvement of those two molecular pathways in ascending axon LTP using just straight pharmacology. So here we block the mGluR1 pathway using an antagonist, and you can see that in this case, the ascending axon EPSC is decreased, and so is the parallel fibers. So on average, although there's a transient potentiation of the ascending axon EPSC, there's no more LTP, but there's a long-term decrease of the amplitude. With only a small relative increase of ascending axon amplitude. Similarly, when we block NMDA receptors with APV, LTP is fully blocked, and both currents show now a depression. So it turns out that in the case of ascending axon plasticity, here, both NMDA receptors and mGluR1 actually seem to work together rather than separately. And they can very well collaborate, in fact, because NMDA receptors can trigger the release of NO, and mGluR1 receptor can trigger the calcium increase that's also required in this situation. So what we don't know at this stage is whether actually both NMDA receptor and mGluR1 receptors are present on the two types of inputs. And we don't know either how they bring about associativity. So associativity means that the a signal coming from the parallel fiber synapses is required to trigger plasticity at the ascending axon. This can be done either through overlap of the synaptic territories or through crosstalk. Now, given the size of the currents we're talking about and the size of the dendrites of a Purkinje cell, actually there's very little chance of reproducible overlap of the two types of synapses. So we really think there is a crosstalk mechanism between the two synapses. What are the possible substrates for this associativity? There are two main options. One is that actually one of those receptors is absent at the ascending axon. So maybe NO or maybe endocannabinoids are not produced and are necessary. And the diffusion from the parallel fiber synapses might be required. That's one option. The other option, which I think is probably more likely, is that the dense parallel fiber inputs actually triggers depolarization, which spreads into the dendrites and can spread to the ascending axon synapses. And there are two ways this could participate. One is by boosting the calcium signal by activation of voltage-gated calcium channels. The other one is by boosting calcium signals through the priming of calcium release from stores. And this has been shown in Purkinje cells, so that's another option. So why do these experiments with inhibition preserved? Usually these experiments are done with inhibition blocked in order to simply isolate the EPSCs. In our case, we have shown that this is actually not necessary to measure the EPSCs. And we know that inhibition is actually important in the system. Granule cells synapse both on Golgi cells and a molecular layer interneurons. We know that postsynaptically, inhibition controls the dendritic potential. So that means it will control activation of voltage-gated calcium channels, for example. Presynaptically also, GABA-A receptors actually control the gradient of chloride in the granule cell axons, and they control the release probability in the parallel fiber synapses. So it is important, and that's why we wanted to do the experiment in these conditions. So we looked at finally at the effect of blocking those GABA receptors to see how important they were. So we did these experiments now in the presence of gabazine to block GABA receptors throughout the experiments. And in this example, you can see that in this case, the ascending axon EPSCs are again increased and the parallel fiber are decreased. And on average, we still observed a potentiation of the ascending axon synapses, yet this potentiation was very variable, in fact, between recordings. And on average, we have no significant LTP. Yet, when you look at the relative amplitude of the currents, it is increased during the experiments. So inhibition seemed to have some importance in the reliability of the induction and the maintenance of the currents, but blocking GABA receptors doesn't wipe out this form of plasticity. The last question we asked was whether the density of the inputs was actually important in this plasticity. So the ascending axon inputs, which are stimulating here, the ascending axon will actually make their synapses all the way up on the dendrites. And so these inputs are sparsely distributed on the dendritic tree. The parallel fiber inputs, on the other hand, is really a beam stimulation. So we're stimulating a dense patch of synapses. So we asked whether the sparse distribution actually made the synapses prone to plasticity. And we did a series of experiments where we stimulate this time parallel fibers, but we stimulate them from the granule cell layer at a distance from the Purkinje cell. And in this case, obviously we will stimulate some ascending axon, but they will bifurcate and make a sparse parallel fiber inputs on the Purkinje cell. So in this condition, again, things are color-coded here, with the sparse input in brown and the dense parallel fiber input in orange. And you can see in this example that both the sparse input and the dense input are decreased following the stimulation protocol. Now, on average, the sparse input was first slightly potentiated and then decreased in a similar manner to the rundown of the synaptic currents. So no LTP of those sparse inputs, but a depression that's not as strong as for the parallel fiber, the dense inputs. So ascending axon LTP appears to be really specific of the inputs and not due simply to the distribution of the inputs on the dendritic tree. So to conclude, I've shown you a new form of ascending axon plasticity with both LTP and LTD of these inputs. The plasticity is associative and it's timing dependent. It's mGlu1 and NMDA receptor dependent. I think it indicates again, suggests again that ascending axon and parallel fiber synapses are really different inputs. Finally, for the cerebellum, I think it could provide a way to functionally associate granule cell inputs and select the local inputs which are linked to the activity in lateral areas of interest. And when I say of interest, it means those parallel fiber inputs which have been selected by the activity of the climbing fiber. I want to thank my collaborators, Rossella Conti at the Université Paris-Cité and David Ogden as well. And I would like to thank you for your attention.
Céline, thank you so much. If you have any questions, please go ahead and raise your hand and I will be happy to call on you. I'll start the question, Céline. So what do you think is the reaction of these synapses to the climbing fiber inputs?
Well, so the only published observation says that there was no modification by the climbing fiber, but when I link it to the climbing fiber activity at the end, because I think climbing fiber is selecting the parallel fiber inputs, which are themselves active, and these are interacting now with the ascending axon. So I think there's going to be a logic in all those, in the end, in all those selected inputs, which is linked to climbing fiber activity.
Any questions for Céline? You noted at the beginning the work on the electric fish, and I'd wonder if you want to tell us a little bit about how you think what you've found relates to that work.
Well, I think in the case of the fish, you have a clear association between motor and sensory feedback. So two different information in order to analyze information and subtract the self, basically. I think the circuit of the cerebellum cannot quite do the same, because here granule cells are going to encode everything. They encode motor information, whether they are ascending axon or parallel fibers, it's the same. They both encode motor or sensory inputs. I think here it's more about associating, and so I guess comparing the very specific input, which might be the receptive field input, with actually all those contextual inputs that are transmitted via the parallel fibers. So the circuit is a bit different, but obviously what's missing here, and it's more difficult to look at, is inhibition, right? We only have half the story here, but I think there's certainly plasticity also at the level of the inhibition. So whether this comes together as a negative image, I don't know, because I don't have the full picture.
Céline, do these ascending axons also synapse on molecular layer interneurons, maybe basket cells?
Yes, they do synapse on both basket and stellate. So most of the time they also recruit inhibition. So when you modify excitation, you're going to also really modify the balance, and you could create a negative image as you recruit inhibition. Right?
Yeah, go ahead. I think there was a question.
Thank you for the talk, Dr. Auger. I was just wondering if in those slices, it was possible to record from other regions like deep nuclei, cerebellar nuclei, and what would your guess be? What happens in terms of ascending axon signal affecting the information going to the nuclei?
So I've never recorded myself from the deep nuclei, and I don't know if the slices are exactly the same. I mean, I guess you're going to have to select a slice that contains the nuclei. You might have to, if you want to have the connection between the Purkinje cell and the nuclei, you might have to take a specific angle of slice. I think the ascending axon is going to modify the activity of the Purkinje cell and the activity of the interneurons. So just like parallel fibers do. So it is going to participate in regulating the output to the deep nuclei as well.
Thank you.
Thank you.
Any other questions for Céline? Okay, well, Céline, thank you so much for your time, and it was wonderful to hear about this work.
Thank you. Okay.